Namaste everyone, I'm Dr. Ankita Sawant and I'm back with a very interesting video on embryonic development of the nervous system. So starting from the scratch. So here we have general embryonic development which are described by the trimester wise. As we know we have three trimesters. First is the foundation of major organs, second is the development of organs, and third is the rapid growth and fully functional organs. So anatomical stages wise we'll see the first stage is the pre-embryonic period, which is from 0 to 2 weeks. In pre-embryonic period, fertilization occurs, blastocyst formation and implantation occurs, then is gastrulation. In embryonic period 3 to 8 weeks, development and differentiation of 3 germ layers into the foundation of the organs. In fetal period, which is from 9 weeks till the birth period, this is the period of growth. So I'll describe you the same by this diagram. Here as you can see, development stage in pre-embryonic period, blastocyst is formed. In embryonic period, the embryo is formed, and in fetal period, the whole-fledged fetus is formed. The events which occur in the pre-embryonic period, in week 1, we have zygote that divides mitotically many times to produce a multicellular blastocyst that gets implanted into the uterus. So in week 1 to 2, zygotes divides in multicellular blastocyst, and that blastocyst gets implanted into the uterus. This is the pre-embryonic period. Whereas in embryonic period, week 3 through 8, blastocyst grows. It folds and forms rudimentary organ system, and that's the reason it is called as embryo. And now in the fetal period, from 9 weeks till 38 weeks or until the birth, embryo is now called as fetus because it has grown now completely. So it grows larger and develops until its organ system is independent, which is not dependent on mother's assistance. Okay. So here are some terminologies. Rostral or cephalic means head, caudal means tail end, dorsal means back, ventral means front. Ganglia are the group of nerve cell bodies. Gyrus, elevation or the crest of the folds on the cerebral cortex. Sulcus, on the other hand, are the grooves, furrows between the gyri on the cerebral cortex. So now we'll see the embryonic development stepwise. In pre-embryonic, first we saw is blastocyst. So the fertilized egg reaches the morula by day 3 and it develops into the blastocyst by day 7, and then it gets implanted to the endometrium. Okay, the fertilized egg reaches to morula, blastocyst, and implants into the endometrium. Take notes guys, please. The implanted blastocyst consists of inner cell mass. This blastocyst consists of inner cell mass which is surrounded by trophoblast. Make a flowchart. The inner cell mass, the blastocyst, differentiates into the bilaminar disc. It has now 2 layers. First is the epiblast layer, which is the top layer of the columnar cells. Hypoblast layer is the bottom layer, and which are the cuboidal cells. Okay, so here day 1, day 2 cell is dividing, day 3 again day 3 and 4 cells are dividing. Cell 8 uncompacted morula, compacted morula. Day 5 early blastocyst is formed, late blastocyst which is hatching, and then finally implantation of blastocyst by 8 and 9. So now this layer is the epiblast layer, this is the hypoblast layer, and this topoblast layer, this layer topoblast. This is the amniotic cavity, this is the endoderm, and re-established uterine mucosa. That was the blastocyst. Now we'll move on to the gastrulation process. The second stage, embryonic period 3 weeks plus. What is gastrulation? The process that establishes the 3 primary germ layers into the embryo. 3 primary germ layers now they are forming here. So it begins with the formation of primitive streak, which is the hollow midline groove along the caudal end, tail end, half of the bilaminar disc. At the cephalic end, tail end, we are seeing the primitive streak. At the cephalic end, the primitive streak is the primitive node which surrounds a small primitive pit. Now the cells of epiblast proliferates, migrates through the primitive pit into the gap between the epiblast and hypoblast. This is known as invagination. Whereas the epiblast becomes, now this is the epiblast, there is this invagination, and this is the hypoblast. So this epiblast becomes the ectoderm, hypoblast becomes the endoderm, and this becomes the mesoderm. This invagination becomes the mesoderm layer. Okay, so this further I explained, this was epiderm, this was epiblast and hypoblast. Here they are formed as the ectoderm. Epiblast is ectoderm and hypoblast is endoderm, and the middle portion, invagination, now has become the mesodermal layer. Okay, so this was the second stage. Now neurulation. What is neurulation? Where the ectoderm around the midline thickens. The ectoderm, which was further the epiblast, thickens to form the neural plate. Now this becomes the neural plate. This neural plate invaginates to form neural groove. It has now neural plate invaginates to form neural groove down the midline, flanked by two neural folds. The notochord, a flexible rod of mesoderm derived cells, define the primitive axis of the embryo. The outer edge of this neuroplate comes closer and merges, fuses to form the neural tube. Neural plates comes closer and fuses and forms a neural tube. Very important note now. Initially this happens around the center of embryo, leaving open neural grooves at both the cephalic and caudal end. So these neural grooves are called as, also known as neural pores, so which closes around 6 weeks of development. If there is failure of closure of this neuropores, this can result in neural tube defects, जैसे कि spina bifida. The hollow part inside the neural tube is called neurocele. Neural tube separates from the ectoderm now, ठीक है, and sinks into the level of mesoderm. Neural tube separates from ectoderm by the neural crest. Okay, I'll explain this whole thing in the diagram below. The mesoderm that flanks the sunken neural tube develops into the somites, which eventually become the skin, skeletal muscle, and vertebral and skull. So the somite is very important now, because this mesoderm layer now sunks the sunken neural tube, which develops into the somites, which will further result in skin, skeletal muscle, vertebra, and skull. Next, some cells on the top of the neural tube differentiates and separates to form the neural crest. Cells of neural crest eventually migrate and give rise to peripheral sensory neurons, autonomic neurons, and sensory ganglia on the spinal nerve. So, as I explained further, this ectodermal layer, this is the neural plate which forms, this comes closer, this neural crust is formed here now, this neural plate is now the crust, this fuses, this is the mesoderm, this is notochord. So these layer fuses, and here this fuses whole, and this is formed here as a neural tube. This is the ectoderm layer which separates from the neural tube by the neural crest. The closure of neural tube disconnects the neural crest from the epidermis or the ectoderm. The neural crest cells differentiate to form most of the peripheral nervous system. Now this neural crest differentiates to form most of the nervous system. Further, as you can see now here, how the development has become. This is the epidermal layer, this is the neural tube, this was the neural crest, which is now the spinal ganglia, this is the notochord, mesodermal derived, these were the mesoderm, which are now called as somites. The notochord degenerates and only persists as the nucleus pulposus. Now this notochord develops into the nucleus pulposus. of the intervertebral disc. Other mesoderm cells here differentiates into the somites. These were here mesoderm, now they are called as somites, the precursors of the axial skeletal and skeletal muscle. What are these somites now? Somites are the mesodermal tissues which develops into the adjacent to the neural tube, and then it develops as the somites, which are eventually called as skin, skeletal muscle and vertebra plus skull. So somites grow in association with developing nervous system, establish early connections. Somites differentiate into three regions: sclerotome, which are becomes vertebral column and skull; myotome becomes the skeletal muscle; dermatome becomes the skin. So hence somites determine the distribution of nervous supply to the mesodermal derived tissue. As you can see in this diagram, this is the sclerotome, which becomes the vertebral column and skull. This is the myotome, which becomes muscle, skeletal muscle. This is the dermatome, which becomes the skin. So that's it for this video. As I wanted to continue, but this will be a very boring video if I continue further. That's the reason I thought of dividing the lecture into the parts. So next upcoming continuation of this video will be the development of neural tube into the spinal cord. We saw till the neural tube has formed. Now that neural tube will form into the spinal cord, that I'll be covering into the next lecture. So stay tuned for that. If you want this PDF, just comment below that I want PDF and I'll be providing you the link.